few moments later. A few moments later.
So much later that the old narrator got tired of waiting and they had to hire a new one.
A few moments later.
Years later.
A very good morning, I'm Dr. Razifah, and today I would like to explain about my lab practical report for BIS3003. So, the title of the lab report is about plant growth and developmental activity, and the objective of my practical report is to study plant growth and its developmental activity, which is how plant grows and develops its roots, leaves, and shoots. So what is actually meant by growth? Growth is the irreversible increase in mass that results from cell division and cell expansion. And what is meant by development? Development is the sum of all the changes that describes an organism's body. So basically there are two types of growth, which are primary growth and secondary growth. Primary growth involves apical meristematic tissues, which can be found on the tips of roots and shoots. Basically, primary growth is responsible for the increase in the length of stem and the length of roots. So in other words, primary growth is responsible for the increase in height of the plants. Moving on to secondary growth. Secondary growth involves lateral meristematic tissues. So this secondary growth is actually responsible for the increase in girth of the plants. In other words, increase in thickness of the root and shoot of plants. So let us learn about the structure of root. So the first thing first, this is epidermis, which is represented by purple color. And the function of the epidermis is to protect the root against mechanical injury, water loss, and infection in plants. So moving on to cortex, which is represented by the yellow color stripes here. So the function of the cortex is to transport materials into the central cylinder of the root through diffusion, and it may also be used for food storage in the form of starch. Next is endodermis. Endodermis can be represented by the blue color here, the blue color stripes here. So the function of the endodermis is to regulate the movement of water, ions, and hormones into and out of the vascular system, and it may also store starch and protect the plant against toxins from moving into the vascular system. So moving on to stele, which is represented by the light orange red color found in the middle part of the root. So function of the stele is to transport water, nutrients, and product of photosynthesis, for example glucose and sucrose, in plants. So moving on to quotient center, which you can find it in the middle part here that contains lots of small stripes. This quotient center is an area in the apical meristem of the root where cell division occurs very slowly, but the cells are able to resume meristematic activity if the tissue around them is damaged. So next, lateral root cap, which we can find it is represented by the dark gray color here. So next about the root cap. Root cap, we can find it here, which is represented by the black color here. So the root cap is actually functioned to protect the root against mechanical injury, as it can secrete lubricating substance known as milky gel that can replicate the movement of the root when root grows by penetrating through the soil. So lastly, it's about columella. Columella is a terminal root tissue with simple tissue organization that can be found in most of the plants, which you can see here represented by the light brown color. So this is basically the structure of the root. Next, moving on to woody stems. As we all know, a woody plant has both primary and secondary tissues. Primary tissues are new tissues formed each year from primary meristems right behind the apical meristem, while secondary tissues develop during the second and subsequent years of growth from lateral meristems, for example, vascular cambium and cork cambium. As I have mentioned just now, there are two types of growth, which are primary growth and secondary growth. So why not just we learn about the primary and secondary growth in a woody stem together? So basically there are two types of meristematic tissues, which are apical meristematic tissues and lateral meristematic tissues. So as we all know, the apical meristematic tissues of stem will form three primary meristems, which are known as protoderm, procambium, and ground meristems, whereby protoderm will mature into epidermis, and procambium will mature into primary phloem and xylem, and lastly, ground meristem will mature into ground tissue, which consists of pith and cortex. So all of these epidermis, primary phloem, primary xylem, and ground tissue are examples of primary tissues. So moving on to lateral meristems, which is the second type of meristematic tissues. So there are two types of lateral meristematic tissues: vascular cambium and cord cambium. Whereby vascular cambium will mature into secondary phloem and secondary xylem, and cord cambium will mature into cord. So all of these three tissues, which are secondary phloem, secondary xylem, and cord, are known as secondary tissues. Next, let us learn about the structure of primary stem. As you can see here, these circular structures is representing the structure of primary stem, and the primary stem is formed during the primary growth of plants. So as you can see here, the purple color structures is representing the phloem, and the function of the phloem is to transport organic materials or the product of photosynthesis, for example glucose and sucrose, from the leaves down to all parts of the plants. Whereas the green region here, the green triangular region here, is representing the xylem tissue. The xylem tissue function is to transport water and dissolved mineral salts from the roots to all parts of the plant throughout the plant. And the cambium, which you can see the straps, the thin straps here, that is sandwiched between the phloem and xylem, will divide repeatedly to form phloem and xylem. And lastly, the middle region of the stem is known as pith. So the function of the pith is actually to transport water and nutrients that is stored throughout the plant. So this is basically the structure of primary stem during is the primary growth of plants. Next, let us learn about the structure of secondary stem. So this structure right here is representing the structure of secondary stem. So as I have mentioned just now, there is one type of primary meristematic tissues known as procambium, which will mature into primary xylem and primary phloem, which can be noticed and observed here, primary phloem and primary xylem. So let us now proceed to one type of lateral meristematic tissues known as vascular cambium, which will mature into secondary xylem and secondary phloem during its maturation process. So as you can see here, secondary xylem and secondary phloem is represented by the red circles and blue circles right here. So there is still another type of lateral meristematic tissues known as cord cambium. This cord cambium will mature into cord during its maturation process. So this is basically the structure of secondary stem. So this is basically the 3D shape of the stem. Okay, so there are two faces of this cylindrical stem, which can be observed on the upper surface here and the lower surface here. So the upper surface here is actually representing the structure of the primary stem. This one, the structure of the primary stem. So similar, right? So yeah, as the stem ages, so it will undergo secondary growth. So and then the secondary stem will then be formed. So yeah, the lower surface here is similar to this structure, right? So yeah, basically this lower surface of the cylindrical stem is representing the secondary stem structures. Last but not least, let us together study the structure of leaf. So as you can see here, the red rod shape here is representing the wet seed cuticle of the leaves. So this wet seed cuticle is actually very important because it can prevent excessive water loss during photosynthesis process. Because especially during hot and sunny day, as it can prevent plants from being dehydrated. So next, you can see this row here represents upper epidermis of the leaves, and down as we go down, you can see this row of cells are representing the palisade mesophyll cells. So as we all know, the palisade mesophyll cells are arranged vertically with each other in a straight position. Why? Because the reason why palisade mesophyll cells have this kind of arrangement is to maximize the rate of absorption of sunlight for the maximum rate of photosynthesis process in plants. So as we go down again, we can see these structures, this irregular shape of structures represent the spongy mesophyll cells. So if compared to pelagic mesophyll cells have regular shapes, and spongy mesophyll cells have irregular shapes. And then spongy mesophyll cells have less chloroplasts if compared to pelagic mesophyll cells. Okay, so let's proceed to the vascular bundle right here, the central cylinder here. This central circle is actually representing the vascular bundle, and as we all know, vascular bundle consists of xylem and phloem, which can be represented by the red color dot and the green color dot. So yeah, and then the vascular bundle is surrounded by a layer of bundle shaft cells. Each single unit here is actually representing the bundle shaft cells. 1 bundle shaft cells, 2 bundle shaft cells, and 3 bundle shaft cells, and so on. So as we go down again, this row represents the lower epidermis of the leaves. And finally, the lower surface of the leaves is covered again with waxy cuticle. And then between the lower epidermis and waxy cuticle, you can find stomata, which is the opening that can be controlled by gut cells during photosynthesis process. So this is basically the structure of leaf.